Good afternoon, Namaste. I, Dr. Mahesh Singh, Associate Professor and Program Chair, School of Agriculture, Galgotia University, welcome you all in NIOS studio. Dear learners, today we will discuss about the major diseases of millets and its management. Under this, we will study about the learning outcome of this is to teach the student about the symptoms, etiology, disease cycle, suitable management practices for important millets and for assured yield. Dear learners, the learning outcomes is at the end of the session you will understand about the importance of the major diseases and their economic losses, their causal agent, the symptoms, what are the favorable conditions to cause the disease and spread the disease, what are the pathogens, what are the disease cycles, and what are the suitable management practices. Under this, you will definitely understand about the major diseases and their practices for suitable management and assured yield. Dear learners, as you know, the Food of Agriculture Organization, that is FAO, has announced at the behest of the Government of India, declared as the 2023 as International Year of Millets. As you know, we are celebrating International Millets Year as 2023. As you know, what are the millets? Millets are the major group of small cereals which are oldest food known to human beings. As you know, these millets are cultivated earlier humans in so many countries, Asian countries, African countries, and other also. But nowadays, after the Green Revolution, you can say we had been shifted to so many cereals like wheat and rice and some other cereals, and definitely we forgot or we less consume these millets. But these have the very importance in as a nutritional value. Small millets are known for their suitability to dry lands, hill area, and as well as less fertile soil. But it provides the staple food for food and grains for millions of poor peoples. You can say as well as the feed grains for the fodder as well as livestock for developing birds. Generally, small millets are extremely tolerant to the drought as well as the and some abiotic stresses also, such as high temperature and poor soil quality. Millets are known for their unique nutritional property, particularly for high fiber content, quality protein, and minerals composition. India's major millets include sorghum, pearl millets, finger millets, which are more commonly known as jowar, bajra, and ragi respectively. And small millets like finger millets, foxtail millet, porso millet, and kodo millets, which are very usually cultivated in our Indian continents. Unfortunately, in the last 6 decades, millets have seen a drop in area despite of Green Revolution in 1960s. Under this diseases are one of the major constant that is limiting factor for reduction of millets production every year, caused by the several entities like fungi, bacteria, viruses, and so many other pests. And these are the some list I have been mentioned here for millets and their regions, cultivated area, and nutritional properties of the millets, and what are the common uses for that. Like pearl millet, that is known as bajra, finger millets, that is ragi, and pearl millet is cultivated basically in Indian region like Rajasthan, Haryana, Gujarat, Maharashtra, Uttar Pradesh, Punjab, and the nutritional value is very high content, very high fibers, protein, iron, magnesium and calcium, and you can commonly see the uses of this is as a roti, that is chapati, and khichdi and porridge also. And finger millets, it is also known as ragi, and it is has been cultivated basically in Karnataka, Tamil Nadu, Andhra Pradesh, Telangana, Kerala, and it have high in calcium, iron, fiber and protein also. You can prepare as for food grains as dosa, idli, porridge also. And next is foxtail millets, that is kangni. It is also cultivated in Tamil Nadu, Andhra Pradesh, Karnataka, Odisha, Maharashtra, Madhya Pradesh, and some other parts of India also. It has a high in protein, fiber and minerals such as copper and iron. Upma and other product you have prepared for this. And next is little millets, that is also known in Hindi as a kutki. It has been cultivated in Karnataka, Tamil Nadu, Maharashtra, Uttar Pradesh and Uttarakhand, and it has been rich in fiber, protein and minerals such as potassium and magnesium. And next is porso millet, in Hindi it is known as China. It has been cultivated in Uttar Pradesh, Rajasthan, Haryana, Gujarat, Maharashtra, Karnataka, Tamil Nadu and other parts of India also, which have high protein content, fiber, minerals such as iron and potassium also. You can make it as a roti, khichdi, kheer and porridge also. And next is barnyard millet, that is sawa in Hindi, and it is grown in Uttar Pradesh, Rajasthan, Madhya Pradesh, Gujarat, Karnataka, Tamil Nadu, and it has also rich in fiber, protein and minerals also like calcium and potassium. You can make khichdi, dosa, idli, upma, etc. And next is kodo millet, which is kodra in Hindi, and Maharashtra, cultivated in Maharashtra, Odisha, Uttar Pradesh, Tamil Nadu, Andhra Pradesh and Telangana. It is also having high rich in protein, fiber as well as minerals such as iron and calcium. You can prepare it as a food like khichdi, pulao, upma, kheer, etc. And next is sorghum millets. You can say it is a jowar, known as jowar, which is cultivated in Maharashtra, Karnataka, Telangana, Andhra Pradesh, Tamil Nadu, Madhya Pradesh and Uttar Pradesh also. And it has also a high in fiber, protein and minerals such as potassium and iron. You can make it in roti, khichdi and porridge. And in spite of this, the importance of the millets diseases, you can see the list of the some important diseases which has been very reduction in their yields as well as their quality of the product. These are the diseases like important diseases like leaf spot, downy mildew, ergot disease, leaf blight, rust diseases, smut diseases, grain mold, blast, bacterial leaf streak, bacterial leaf stripe and bacterial stalk rot, anthracnose and mosaic diseases. Dear learners, we will study about the ergot or sugary disease of Bajra. As you know, pearl millets or Bajra is grown and widely in African as well as Asian country. The disease has been severely reported in Ghana, Nigeria, Somalia, Telangana, Uganda, Zambia, Zimbabwe, Pakistan and some other part of the world where Bajra has been cultivated. In India, the disease is known to occur in Uttar Pradesh, Haryana, Maharashtra, Rajasthan, Karnataka, Tamil Nadu, Andhra Pradesh and Uttarakhand in moderate to severe form. In severe infection, crop losses up to 70% have been reported due to the ergot disease. Dear learners, ergot infection causes loss in the seed as well as the quality of the seed, their germination percentage and the seedling emergence also. During 1967 to 78, the disease broke out in epidemic form on newly imported hybrid Bajra variety in Maharashtra and Karnataka. The pathogen produce mycotoxin, as you know, it is called ergotoxin, which causes disease in human as well as in animal, known to ergotism by the consuming of the infected grains. Dear learners, these are the causal organisms of the ergot disease. Like the ergot disease causal is it is a fungal pathogen which is known as Claviceps fusiformis. And the symptoms, we will discuss about the major symptoms of this disease. Like first symptoms is honeydew stage of the this symptoms. In these symptoms, you can see here in the photographs, pinkish or light brown honey colored sticky fluids exudes you can say appear from the infected spikelets. The inflorescence completely become damaged sometimes at the over infection, and honeydew contains conidia of the fungus. And the next stage of this symptoms is small dark brown sclerosial bodies are formed on the ears. These sclerosia replace ovary as well as the seed grain. These sclerosia, that is called ergots, they contain alkaloids which is responsible for the ergot poisoning in animals. You can see here in photographs the damage of the ear head of Bajra. The grains has been completely damaged due to the infection of the ergot. And the pathogen is basically soil borne, as well as sclerosia admixtures with the seeds, and infection is basically in airborne condition. Means you can say the disease spread spread through the soil as well as seed and air condition also. How the pathogen survive throughout the year, dear learners? The fungus survive through the sclerosial bodies in and conidia on it. The previous year's crop debris has been infected and fell down on the ground, and the pathogen carry out through seed also. And when the next favorable conditions come in next year, the pathogens get spread and as well as germinate and the cause the primary infection. The primary infection caused by the ascospores and the conidia present on the sclerosial surface, as you have already seen in the previous slide, and through the stigma. And the secondary infection caused by the conidia produced by the contaminated in honeydew like exudes, and secondary spread occurs through the conidia by insect or rain to the healthy plants. And the dear learners, you can say when the rainy seasons come, the infected part of the Bajra to healthy ones, and after that the infection get start in this is called secondary infection also. High humidity favor the high humidity at the 85 to 95% if the relative humidity infected to healthy one also. And cloudy weather is also favor, and sunshine with the help of rain splashes also to increase the disease severity. Dear learners, this is the disease cycles as I have already discussed to you. This is the infected portion of the plant debris through infection, the germination of the conidia and spread through the soil as well as through air and seed conditions also. And now about the disease management. What are the major practices by which you can disease management? We have studied here. We will study here. Like first one is cultural practices. Under cultural practices, since this is the pathogen is basically soil borne, so that you have to follow the long crop rotation. crop rotation for a long time. You can use the crop rotation for cultivation of the one crop after the 2 to 3 years gap. Use of the sclerosia free seed for the cultivation, and deep plowing in summer season. In photograph you can see here the deep plowing has been done by the tractor drawn machine, and this practices should be done in the month of basically in May and June, in means summer season. Dipping in the seed in the 20% brines, or you can say salt solution, and this floats the sclerosia, which can be removed after the floating, and intercropping or mixed cropping you can also favor with the help of mung bean. You can say adopt early sowing before 15th July, which is favored to less disease, and destroy the grass host before the sclerosia formation. Dear learners, these are the some resistant variety. You can also grow like ICI 7517S1 and SSCPS 252S4 shown high level of resistance against ergot disease. Some chemical spray are also listed here. You can apply like Xyrum at the rate of 0.2%, or copper oxychloride or Zinab at the rate of 0.2%, or sulfur 0.25%. And you can also apply Bevestine at the rate of 0.1% just before earhead emergence, and you can repeat it 2 to 3 times depend upon the severity at 7 to 8 days intervals. Dear learners, downy mildew. Now we will discuss about the downy mildew. Next disease is downy mildew, or you can say green ear of Bajra. The distribution and economic importance of green ear of Bajra is this disease was first reported from India by E.J. Butler in 1907. Now it has been reported as a major disease of all Bajra growing areas of the world, basically in Asian and African countries. It is locally known as Zogia Rog in Hindi, and it causes loss up to 30% in high yielding varieties in India basically, and the pathogen is the obligate parasite. You can say it is a kind of biotroph, and the disease is severe in ill-drained or low-laying area where drainage facility is very poor, and the causal organism and symptoms of this disease is the causal organism is Sclerospora graminicola. The disease symptoms, you can see here in photographs, there are some leaves, infected leaves. You can say there are 2 stages of symptoms, like first one is downy mildew stage. The symptoms of downy mildew is appears as a chlorosis on the first to third leaves of the seedlings. White downy growth appears on the lower surface of the chlorotic area. Means basically when the Bajra is grown in the field and the lower surface, means the soil is contaminated by the previous crop debris or the pathogens, then lower leaves get infected by the pathogens, and the adjacent the other leaves will also contaminated through that. That's why the downy growth and whitish patches you can see here in the photographs of the leaves. And next stage is a green ear stage, and the transformation of the floral organ means after the infection of the leaves, the inflorescence get also infected by these pathogens. The transformation of the floral organ into the twisted leafy structures, that is called phyllodes. You can see here in the photographs, this is the ear head of the Bajra, but it is not a normal Bajra ear head. You can see here in photograph photographs in the lower Bajra earhead, the leafy structures have been clearly seen by you, leaves are small, very small scaly leaves are seen here, and other part is normal, and the inflorescence is also malformed like that. This gives an appearance of the green leafy mass, hence the name, it is called the green ear. The bristle of the spikelets become hypertrophied, means the hypertrophism you can see here, and the inflorescence get damaged, and sometimes it becomes sterile in form, and sometimes only a portion of ear shown, the green ear, and other symptoms is rest symptoms is a normal, means you can see here the other 50% or some part of the ear head is looking just like a normal ear head, but some part is infected due to the green ear, and the leafy structures have been also seen by you clearly. And now about the disease cycle. Dear learners, disease cycle is very important to understand you to manage the disease, means how the pathogens survive and how the pathogen is spread in the field, and what are the means by which the pathogens come or rotate through year by year. That's why you have to clearly understand about the their survival condition, their perination, their mode of spread, as well as their means all complete disease cycle. First one is perination or survival. The pathogens survive through oospore in the disease part, fall down on the ground, as well as through the mycelium in the infected seed, that is basically internally seed borne. Dear learners, the pathogens survive through oospores in the fall down, means the infected crop debris fell down in the field, and that infected parts mix with the soil. After mixing the soil, the oospores survive throughout the year as dormant conditions. When the next year favorable conditions come, the pathogens get germinated, and you can see the seed is also responsible for the and as a internally seed borne, means pathogens survive through seed also, and when the seed sown contaminated seed sown in next year, the pathogen get easily contaminated and germinate in the next cropping season, and the primary infection get started. And collateral host like Panicum species also play a very important role for perination of the pathogens. Dear learners, there are some collateral hosts also like Panicum species which are responsible to perinate or disperse from one cropping season to another cropping season of the pathogens. Dear learners, now in continuation, the primary infection, how the mode of the primary infection get start. The oospore present in the soil germinate and cause the primary infection. As I told you, dear learners, means primary infection survive, the pathogens survive through dormant condition in the field, and when the favorable conditions come, means favorable conditions like favorable temperature like 25 to 28 degrees centigrade and high humidity, the pathogens germinate, and the seedlings also get affected around 9 days after the crop sown. The infection is spread upwards systematically along with the plant growth. Means the basically the pathogens get affect the lower surface of the leaves at the time of germinating, which is contact with the soil, and after that the pathogens get upward direction, move through this disease. Now we will meet after a little break.
जीवन ये प्रकाशित करने, राहों को आलोकित करने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
जीवन ये प्रकाशित करने, राहों को आलोकित करने।
की सीमा से विद्यालय जो मुक्त हो,
संसाधन समय की सीमा से विद्यालय जो मुक्त हो।
कभी पढ़े हम कहीं पढ़े, वे विषय की लोट से युक्त हो।
कभी पढ़े हम कहीं पढ़े, वे विषय की लोट से युक्त हो।
मित्रों हम उठे और जागे,
मंजिल आने की ध्वनि सुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने।
जीवन ये प्रकाशित करने, राहों को आलोकित करने।
राष्ट्रीय मुक्त विद्यालय का है द्वार खुला सबकी खातिर।
शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर।
शिक्षा परीक्षा का अवसर एक बार ही क्यों मिलता फिर। दुनिया में इसके केंद्र बात,
कोई मन चाहा केंद्र चुने। हम अपना दीपक स्वयं बने, हम अपना रास्ता स्वयं चुने। कोई मन चाहा केंद्र चुने।
Dear learner, welcome back once again. We are discussing about the green year of Bajra, and under this we are discussing about the disease cycle. And disease cycles, as I have already told you that the primary infection and survival also. The primary infection, the ooze spores present in the soil which has been germinates and cause primary infection of underground parts of the seedlings around 9 days after the crop sown. Means the lower leaves has been easily affected by the contaminating soil, and simultaneously the infection is spread upwards systematically along with the plant growth. When the plant growth in the height, then the disease also moved upward in direction. Dear learners, the secondary infections is also responsible by the caused by the ooze spores produced by the germination of the sporangia, which has been the dispersal of the sporangia or takes place by means of winds, by means of water as well as insect to healthy plants. Dear learners, the winds are also responsible for the dispersal of the pathogens, when the winds blown, the pathogens also move for a long distance, and water is also responsible through irrigation water, through rain splashes, as well as insects are also get infected, means the insects sit on the infected part of the plants, and they also move from the one infected plant to another healthy one. The pathogens attach with their body, their mouth part, as well as their legs also, and they move from one place to another place also, and one infected plant to another healthy one also. For predisposing factors, the high relative humidity, that is more than 90%, and low soil temperature, that is you can say if the soil is dry conditions and the temperature range is about 20 to 25 degrees centigrade, are the most favorable conditions for the spread of this disease. Dear learners, these are the favorable conditions which has been basically in the month of July and August. At that time, the disease has been severely spread and affect to the Bajra plant. And now about the disease management. As you know, cultural practices is very important for the managing of any kind of disease. As you know, the pathogen is a seed as well as soil borne. That's why you can use the disease free or you can apply the certified seed for the sowing purpose, and you can destroy the collateral host which are responsible to harbor the these pathogens. So you have to destroy and collect that kind of collateral host, and you can dig a pit and you can submerge in that, and you can remove and destruct the infected plant parts in the early stage also. You can avoid the monoculturing process, don't grow the Bajra regularly in the same field for a long time, and you can follow the deep plowing in summer season. In previously I have told you, means deep plowing has been implemented basically in the month of summer season, and that's why all pathogens and as well as the weed seeds also submerge in the soil and get exposed through high temperature of direct sunlight. That's why so many pathogens are directly killed. Early sowing should also be followed. That's why the major severity conditions you can break, and the intercropping with the mung bean also reduce the disease incidence. You can avoid the waterlogging in the field conditions, and these are some cultural practices which are very important for the managing of the this disease. Now about the resistant variety, you can grow the hybrid variety like ICMH 88088. It has been produced by the Acrisat and the Pusa 23, Mallikarjuna, and ICMH 451, etc. These are some resistant cultivars you can grow in it. And now about the chemical control. As the pathogen seed as well as soil borne, so you have to treat the seed for the chemical fungicide like Apton SD35, that is known as metalaxyl, at the rate of 6 gram per kilogram of the seed. You can also treat the seed by using the mancozeb at the rate of 2 gram per kilogram of seed, or Thydam at the rate of 3 gram per kilogram of seed. This gives, you can also treat the seed with the help of hot water, hot water at the rate at the 55 degrees centigrade for 12 millets, and you can also after that you can dry in the shade condition. and steeping or soaking the seeds in 0.1% mercury chloride solution. You can say the Chlorrex at the rate of 2.6% for 2 minutes only, and followed by the 3-4 washing with the tap water, and treat the seed with the PGPR, plant growth promoting rhizobacteria like Pseudomonas fluorescens, Bacillus subtilis, and you can spray the mancozeb in the infected crop at the rate of 0.25%, or you can also apply the Ridomil MZ 72 at the rate of 0.25% within 1 month of the sowing. Dear learners, now about the grain mold or head mold disease. The distribution and economic importance of grain mold and head mold disease is sorghum is a genus of about 25 species of the flowering plants in the grass family. Some of these species are grown as cereals for the human consumption, and others are used for the fodder purpose. As you know, sorghum, so many varieties of the sorghum has been widely used, cultivated in India as abroad also for the fodder, as well as some variety for the human consumption also. Grain mold is a major disease of Kharif season basically, and it is common in many countries like Asia, Africa, North and South American country, and the production losses due to the sorghum grain mold range from the 30% to 100% depending upon the cultivar, time of flowering, as well as prevailing weather conditions during the flowering to harvesting condition. Now about the pathogens. Dear learners, the pathogens, that is a causal organism. The disease is caused by a group of fungus like Fusarium, Curvularia, Alternaria, Aspergillus, Chitomium, Rhizopus, Helminthosporium and Phoma. These are the fungal pathogens which are responsible to cause this disease. And now millets host means the host range is sorghum, pearl millet, finger millets also get affected by this disease. You can see here in photographs the pathogen is get affected by the seed of the seed grains also. Now about the symptoms of the pathogen. Symptoms depending on the fungus involved in the grain maturity stage and the severity of the infection, symptoms could be highly variable. Severely infected grains is fully covered with the mold. Particularly infected grains may look normal to discolored also. Grains show symptoms of the fungal infection and develop fungal blooms of various colors like black, white or pink. Depending on the infection of the infecting the fungus, these show the color as well as various blooms colors also. The infected grains are light weighted, soft, powdery, look like powdery appearance, low in nutritional quality, poor in seed germination, as well as the market value is very low, and it for the human consumption. So that's why you have to understand deeply what are the major factors or what are the symptoms, what are the suitable management practices to manage such kind of disease in sorghum. Now you can see here in photographs the symptoms has been clearly shown in the infected grains of the sorghum, and the sorghum is showing the seed showing the black of molds, means fungal appearance on the seed grains also. Now about the you can see here the different seed is affected by the sorghum grains by these pathogens, means whitish, brownish, blackish appearance of the fungal pathogen. Now about the disease cycle. The disease cycles mean the pathogens, survival of the pathogens. The fungi survive as parasite as well as the saprophyte in the infected plant debris. Mean the previous year's plant debris if get affected, the pathogens survive as a parasitic as well as saprophytic conditions. That's why this is the very challenging to manage this kind of disease, because the pathogens survive as a dual mode, means as a parasitic as well as saprophytic in nature. The fungi mainly spread through airborne conidia. Airborne conidia germinates and is spread through one plant to another healthy one. Rain also helps to spread the pathogen as well as reaching their severity during the flowering and grain filling stage. The favorable conditions for the spread and germination of the disease is wet weather during the flowering, that is favors the grain mold development. The longer the wet period, the greater the mold development. If the wet period is very long, the mold infection is very high. That's why you have to understand about the weather and favorable conditions by which the pathogens survive and spread from one place to another place. The compact ear head are highly susceptible also, means if the ear head is loose, the pathogen is less susceptible, but the compact ear head of the sorghum is responsible to highly susceptibility. And now about the disease management. First of all, you have to adjust the sowing dates to avoid the warm and humid condition during the flowering to grain maturity. Spraying of the any one of the following fungicide, like you can during in case of the intermittent rainfall during the ear head emergence, and also at the milky stage like mancozeb at the rate of 1 kilogram per hectare, or you can also apply the Capton at the rate of 1 kilogram with along with the arufensin at the rate of 100 gram per hectare. These chemicals are responsible to kill and spread the further spread of the pathogens in the entire field. Dear learners, now about the blast disease of millets. Blast disease is also very important disease which are economically important, means this loss cause very losses to the millets, and distribution and economic importance. We are discussing about the finger millets and their finger blast of the finger millets. The particularly known as ragi, means finger millets is also known as ragi or mandua in India, and it is an important crop used for food, forage as well as industrial products, and grown under the wide range of climatic conditions generally cultivated in tropical and subtropical regions of the world. The disease was reported for the first time in India from Tanjore Delta of the Tamil Nadu by Macrae in 1920. Since then, the disease is known to occur almost every year during the rainy season and in all major ragi growing area of the country. The grain yield loss due to the blast disease could be over 30 to 80% depending upon the severity and climatic condition. The blast millets, that is the host range is pearl millet, finger millets, foxtail, barnyard millet, porso millet, and little millets get also affected by this blast disease pathogen. Now about the pathogens and the symptoms. The causal organism is Pyricularia oryzae, that and the perfect stage of it is Magniparthigracia. The symptoms of this disease is the plant is susceptible to the disease during all stages of its growth, like from seedling to grain formation stage. means any plant stages of the plants get affected by this pathogen, and the small alloys spindle shaped lesions are formed at the initial stage, and later on this spread is spread through collapse together to form the greater and bigger stage. You can see in the photographs the small alloys spindle shaped lesions are formed at the initial stage, but after that initial stage it becomes a boat like structure and increase in the size as well as the become in the larger lesions and get collapse together to form the cover the greater area. The distal portion of the leaves beyond the lesions may hang and drop off. After that, after the severe infection, the leaves become hang down and drop off. Stem infections causes blackening of the nodals also, nodal region basically, and nodal region get very damaged, and after that they can easily break down. Maximum damage is caused due to the neck infection only. When the neck infection of the finger millets gets affected, then the maximum in damage may help. During the severe infection, the ears hang down from the stalk and sometime may also break away. Means the if this when the severe infection may held down, then the whole ear head break down and the your whole crop has been damaged. Means the your production is drastically reduced by that particular plant. Now about the disease cycle. The pathogen harbors in glumes, straw as well as in the some germinaceous weeds. The seeds have also been found to carry the pathogen in the pericarp as well as in the endosperm. The climatic conditions having the temperature is about the 25 to 30 degrees centigrade and more than 90% of the relative humidity which are responsible to spread the disease, and with the help of cloudy days are also intermittent rainfall between that days will increase the disease severity. Continuous rainfall time to time heating may lead to the heavy losses of the crop. It also has been observed that the incidence of the blast disease increases with the increase of the doses of the nitrogen. If you have applied the maximum doses of nitrogen, means the application of the nitrogen doses, the plants get very soft and become easily affected by the kinds of so many pathogens, and your crop is get very easily affected by this disease also. Now about the disease management, since the primary infections comes from the seed-borne inoculum, treating the seed with the triconazole at the rate of 8 gram per kilogram of the seed. During the later stage, the crop to spray of the propiconazole at the rate of 0.1% or carbendazim or triconazole at the rate of 0.05% or to spray of the SAF, means at the rate of 0.2%. And at the time of starting of the flowering, and again 10 days later, and also you can find the effective for the reduce of the blast disease. Now in continuation study, for further study you can refer these reference for your further and more study. Thank you very much to all dear learners for your patience learning. Thank you very much.